In this video, we're going to take a look at how to draw proper biological diagrams. A biological diagram just means a drawing of what you see under the microscope. We're going to see the different rules for biological diagrams, and you do have to make sure you follow them because you will be assessed on the degree to which you follow those rules. We're going to come back to this slide in a minute. We're just going to go take a look at the sample drawing that you'll see over here. So, the first thing is that you'll have to draw the diagram on a piece of white paper, landscape. Landscape, okay, not portrait, landscape. You'll have to have a title for your specimen. It should be in the middle and it should be underlined. Usually it's just a specimen that you're observing. So you might be observing a onion cell, or you might be observing a paramecium cell. So it's usually written on your slide or somewhere in your lab. You'll see what you're observing. You write it out, underlined. Then you draw the specimen that you see to the left of the page, not in the middle, to the left of the page, not to the right, to the left of the page. And you should draw that in pencil. Everything is done in pencil. No pen, pencil. No coloring crayons, pencil. No markers, pencil. You'll then label parts of the specimen. When you label parts of your specimen, you should label to your right. These should be a bit more to the right side. So label to the right. So what you do is you make a line that goes from the part you're trying to label all the way to the right side, and you name that part. Don't put the word on the line. It should be at the tip of the line, more towards the right side. What's important is that your labels all end roughly at the same spot. So these lines should be a little bit longer, roughly at the same spot. Okay. Your line should never cross. So this line should not go through here. This line should go up here, and then you make it horizontal after. So never cross your lines. And make sure that the lines actually touch what you're intending to label. If it's trying to label a membrane, make sure it's touching that membrane. On the bottom right, you're going to have your drawing magnification, and we'll teach you how to make a drawing magnification shortly. So, remember, as I just mentioned, your drawing has to be on paper that's 8.5 by 11, white paper. It has to be in landscape form, not portrait. You have to have a title that's in the center and underlined. You can underline it. You should underline it with a ruler. Your drawing should be large and to the left. It should take up a big chunk of your page. It shouldn't be a small segment here. It should be a large part of your page taken up by the drawing. To the left side, not in the middle, not to the right, but on the left. You want to make sure your drawing has firm, solid lines. You're not going to sketch. No sketching like you would do if you were an artist. Just firm, solid lines. You're not going to shade anything in. If there's any dark spots, you don't shade it in. Instead, you stipple, which is where you make repeated dot motions. Dot, 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 dot. We'll see that in a minute, what that looks like. So you're going to stipple. And at the bottom right, you'll have the magnification, your drawing magnification. Whenever you are making your labels, they should go to the right, and you need to use a ruler, and they need to end up at roughly the same spot. And make sure your lines never cross. Make sure they're always parallel to each other, and they end up roughly at the same spot. And again, these I would probably want them more to the right than what you see over here. So you can see over here some examples of what not to do and what to do. You can see here when you're labeling that lines have crossed here. That's wrong. You never want the lines to cross. So make sure your lines don't cross. Over here you can see something that has been sketched. You don't want that. You want something with firm, solid lines. No sketching, just firm, solid lines. And over here you'll see something was shaded in. It was colored in. You don't want to do that. You want to just make little dots. So dot, 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 dot to show shading. That's called stippling. Okay? So no shading in, you do stippling instead. So we're going to take a look at how to calculate the drawing magnification. In the previous video, we saw how to do the actual size of the cell. You have your diameter field of view, how many times your cell fit across, your diameter field of view divided by how many times your cell fits across, and that's how big your organism is. Now, when you draw your specimen on a piece of paper, the drawing will look larger than what the actual specimen is. So you would calculate what's called the drawing magnification. How big is your drawing compared to the actual specimen? So if I had an ant, I'm going to say let's say I have a big ant. Let's say it's a 1 centimeter long ant. And I draw a picture of that 1 centimeter long ant, but I make it 15 centimeters. Well, if I ask you what my drawing magnification is, you would do, well, the ant is 1 centimeter, the drawing is 15, you do 15 divided by 1, the drawing is 15 times larger than the actual ant. So that's what a drawing magnification means. So to do that, what you have to do is, you have to figure out the actual size of your specimen as we saw in the previous video. You would then measure the size of your drawing. You would then do the drawing size divided by the actual size of the specimen, and that would give you your drawing magnification, which is what you put at the bottom over here. We're going to do an example together, but it's going to be an important calculation for you to do. So, we're going to do an example here. Here we have a drawing of a protist cell. This cell is being viewed under medium power. So this is you viewing the cell under medium power with 1500 micrometer field of view. You want to figure out how big your drawing is compared to the actual specimen. So first you need to find the specimen size. So remember, you're a medium power field of view, so that's 1500 micrometers or 1.5 millimeters. You have to estimate how many times this fits across the field of view. So maybe it goes across 1, 2, 3, maybe 3.5. You could have said 2. That would have been fine. And I'm measuring lengthwise here because I measured my drawing lengthwise. Okay, you have to make sure you're consistent with how you're measuring. So to find my cell size, I'm going to do my 1500 divided by 3. That's 500 micrometers. Or if you want to find it in millimeters, you divide by 1,000, 0.5 millimeters. Then I'm going to measure the drawing size. The drawing is about, so here we have 1, 2, 3, 4, 5 centimeters, 6, 7 centimeters long. Now, my unit for the specimen was micrometers. So when I do my magnification calculation, drawing divided by actual size, the units have to match. So if I have 7 centimeters, I can convert that to micrometers by doing centimeters to millimeters is times 10. And then millimeters to micrometers is times 1,000. So essentially from centimeters to micrometers, it's times 10,000. You can do it in a series of steps or in one full step. But I would do 7 times 10 to get to millimeters, and then 70 times 1,000 to get to micrometers. And so now I have micrometers and micrometers here. And so the drawing size is 70,000 micrometers divided by the specimen size, which is 500 micrometers, which means 140 times. So the drawing is about 140 times larger than the actual specimen. And that's what I would put in to my drawing magnification area on the bottom right of the page. Some students might have converted this to millimeters instead. So they would have done 500 divided by 1,000 to go to millimeters. And they would have simply done 70 divided by 0.5 to give them 140 times magnification. So that's called calculating the drawing magnification. And that's what would go at the bottom of your drawing as seen over here. So in this video, we saw how to do biological drawings. We saw how to calculate drawing magnification. We're going to finish off by taking a look at another labeling example. This here is a plant or animal cell under an electron microscope, which is a very powerful microscope that uses electrons. And you can see a lot more organelles and features. So I want you to figure out if it's plant or animal, and then I want you to label some of the parts. So this one is a bit tricky, but you can take a look at some clues. You'll notice that you have an outer part labeled and an inner part labeled. So that means we're probably trying to indicate that there's maybe a cell wall and a cell membrane, making this a plant, probably. And so if we look over here, this is probably your cell wall. This over here is probably your cell membrane. Now, this part here looks like a chloroplast. If you were thinking that, you were correct. Cell wall. This large organelle, knowing it's a plant now, is your large central vacuole. This is your cell membrane, sometimes called a plasma membrane. This is hard. This one's really hard to tell. It's called a Golgi apparatus. These ones I would say are fair for you to get, but this one was a bit tougher for you to get. This would be a mitochondrion, and the reason that you could not put a vacuole here because there's a large central vacuole for a plant, and you could not put a lysosome because that's a large, because plants don't have lysosomes, so mitochondrion is your next best guess. This over here, it's the second largest organelle in the plant cell, command center of the cell, it's your nucleus. But once you know it's a plant, things start to make more sense. As soon as you hear plant or figure out it's a plant, this looks like a chloroplast. This looks like the cell wall and plasma membrane. This one's tougher, so that's not something I may ask, but it's always a good thinking task for you to try out to figure out what this could be. If it was darker, you could have seen it in more detail. Maybe you can try to figure out why it's a Golgi body or Golgi apparatus. You can try this one out over here. Try to guess what type of cell it is. So if you pause the video and you tried it, you're probably guessing that it is an animal cell, and there's a few clues given out. First of all, you can see that there's only a cell membrane or a plasma membrane. There doesn't seem to be a cell wall, and you can try to label the other parts now that you know that it's an animal cell. And here we didn't learn these parts. These are called centrioles. But you can take a look at this over here. Knowing it's not a plant cell, this may be a mitochondrion, so 2 mitochondria. This large organelle here, probably the nucleus. This is hard to tell. It could be a lysosome, could be a vacuole, could be a vesicle. Again, Golgi body. It's a bit easier to see what it is here in that situation. And in this case, this ends up being a lysosome. So with more context, you're better able to label these parts of cells. Now, we won't be able to see this many organelles under the microscope that we have because we don't have electron microscopes. We have compound light microscopes. We can probably see things like the nucleus, for example, but not much more than that. So now that we've gone through the entire lesson, you should be able to draw a labeled picture of a microscope and summarize what parts do, explain how to hold a microscope with a drawing, explain how to locate specimen at low, medium, and high power properly, and explain how to put a microscope away as well, explain how to calculate total magnification, estimate the cell size, how to calculate drawing magnification, and do a proper biological diagram as well with all the proper rules. So it would be a good idea for you to take a look at some examples and make sure you follow those examples when you're trying to do your biological diagrams. One more important thing I did forget to mention earlier too is when you're doing your biological diagrams, it's important that you draw what you see. A lot of students will look at the name of the specimen, go to Google, and then try to draw what they see on a Google image. And I'll immediately know that they didn't draw what they saw because the microscope actually couldn't see the details that the Google image provided. So make sure that you're drawing specifically what you see under the microscope, not what you think you see, not what you Googled. It must be what you're looking at under the microscope.